Our goal is to emulate that process, to make a cut in a gene of interest, and then to shepherd that into a repair pathway of our choosing. And at that point, we begin to diverge from this example of the immune system, because first of all, we like a gene that would be useful to modify. And well, we thought we'd cure cancer with this, right? We just inactivate the oncogenes. And people told us, well, that was, maybe it's not the place to start. Maybe we should start with single gene disorders. And we are told that there are about 5,000 of these single gene disorders, some of which affect just a few patients worldwide, and some of which are quite prevalent. And in Dr. Bloomberg's talk, he actually mentioned one of these, which is sickle cell anemia. The interesting thing about this is that this same mutation has recurred 4 times independently in human evolution. So all patients have the same mutation. There are about 90,000 patients in the US, many more worldwide. It is a bad disease. You'd definitely like to cure it. It's monogenic. There's a good animal model, and there's an accessible stem cell. So it has a lot of the characteristics of a good place to start. Our device will involve incising the sickle cell gene. In this case, we'll use synthetic nucleases. We thought of using the RAG endonucleases, but they're not structurally well understood, and it just wasn't, we thought, the way to go. So instead, we're going to use zinc finger nucleases that we already have. And we're exploring delivery by transferrin receptor-mediated endocytosis. The idea is that we'll create a double-strand break near the sickle cell mutation, or in principle, any mutation for a monogenic disease. We will engage a specific repair pathway. In this case, it will not be non-homologous end joining. It will be the other double-strand break repair pathway that involves template-dependent synthesis, initiated by binding not of the Q protein, but of a protein called RAD51. And that filament then invades a donor template. And so this is now a modification of a machine that does not involve adding one component, but adding 2, at minimum, the zinc finger nuclease and the donor template. Where are we with this? We have the zinc finger nucleases for globin and also for some model genes. We are working on transferrin receptor-mediated endocytosis. The challenges are efficiency and specificity. We'll use our real-time visualization capability. We need to be able to monitor and suppress mutations and rearrangements that might be introduced inadvertently. And an ideal gene correction machine would be independent of foreign DNA and proteins, non-immunogenic, and amenable to temporal control. We are exploring ideas for photo control of the process. I want to acknowledge my collaborators, Gang Bao at Georgia Tech and the other members of our center, as well as the people in my own laboratory that are working on this, and tell you that I've covered 4 topics: double-strand breaks in the response, imaging that response through modified components of the machine, inhibiting the response by adding 1 additional component, and the idea of re-engineering the repair machine by adding 2 components, a nuclease and a donor template that will shepherd this into a recombination pathway. Thank you very much.
Thank you for that fascinating presentation. Are there some questions, please? Your name and affiliation.
I'm Tad Hogue. You mentioned the second mechanism of DNA repair uses a template. Mm-hmm. So in the normal use of that, where does that template come from?
It could be the homologous gene on the other chromosome, or it could be a sister chromatid. These 2 mechanisms work in sort of a yin-yang way. The non-homologous end joining is an acute response to things like a radiation injury. The homologous recombination seems to be more important in genome maintenance because people with, well, BRCA2 patients would be an example of people with a sort of a subtle defect in that makes them susceptible to genetic instability and cancer.
Question?
Kester, Penn State, excellent talk. So one of the double-edged swords of radiobiology is it does kill the cancer, but it also kills the endothelial cells, and you therefore have trouble adding secondary drugs, or in your case, transferrin-mediated small fragments of antibodies. Is there some way you can take your technology and gear it to a targeting approach so that the transferrin receptor-mediated antibodies only go to the cancer cells and not the endothelial cells of the nascent vasculature?
Well, maybe, I don't know, maybe it's a triple-edged sword. So sometimes you want to kill those endothelial cells. I've talked about this as if you're killing the tumor cells, but in fact, there's some debate whether, it's the tumor cells or some of the other cells that are associated with the tumor. Assuming that we're killing the cancer, it is not clear at all that normal tissue damage and killing the tumor work by the same pathways. In particular, killing the normal cells is probably not working by post-mitotic cell death because the cells either aren't dividing in the first place or they have intact cell cycle checkpoints. So I think you can make an argument that radiation works anyway and that we are exploiting the natural differential and just increasing the differential. And then, of course, with any nano approach, there is also the possibility of adding a layer of targeting. So I gave as an example the folate receptor because it's used in tumors, but I think we can imagine a lot of other receptors that you might want to target for your favorite cancer. I'd also like to say we think this endocytosis of the free protein is going to carry us at least as far as some of the preclinical studies. But clearly there's a lot happening with nanoparticle delivery, as you told us yesterday. And so I don't know that that'll be the final device. I think there's plenty of room at the bottom to develop other devices.
I'd like to make a comment. You referred to the early genomes in trying to explain the big mutational change that took place. One of the things that's really possible now, because of the assembly of so many genomes in one place in a database, is kind of comparisons, ready comparisons of genes. And if we want to look at other functions of our genes, a very useful thing to do is look at the homologous genes in very early species. Because many of those effects that were useful to the earliest species in the evolutionary story are actually still present in our genes, and probably some modified form. So a systematic study of genes it's now available. You can look at them and see what they are.
So I have to tell you why it wasn't obvious that this system was present in other organisms. Some organisms have lost it. And in particular, E. coli has lost this whole system. It gets by with other repair pathways. And for a long time, at least somebody like me, maybe thinks E. coli is the only bacteria that exists, and it didn't have it, and so it must be specific to higher organisms. Well, it turns out lots of other eubacteria have the pathway. I am not sure whether it's turned up in the archaeal genomes yet or not, but the prediction is that it would, because it's clearly ancient.
One of the things that we encouraged in the astrobiology program was the study of extremophiles. And I think it really helped move the field forward a good deal, and many more are now unknown and understood.
So we've passed the 1,000 genome barrier, and I guess we're someday maybe going to reach the 10,000 genome
I'm sure that'll
level.
happen.
Most of these are single-cell microorganisms.
Well, thanks again for an excellent talk, very stimulating, and we'll go on to our next speaker.